Okay, continuing with Fundamentals of the Nervous System, Chapter 11, Part C. Okay, so where neurons come together, it's called a synapse, and it follows all the same rules that we talked about in the muscular system with the neuromuscular junction, which is also a synapse. So the synapse is the junction that transmits information from the neuron, presynaptic neuron, to either a postsynaptic neuron or to an effector cell, a gland or smooth muscle or a cardiac muscle or skeletal muscle, it doesn't matter, some sort of an effector. So the presynaptic neuron is the one before that conducts the impulses to the synapse. The postsynaptic reacts to that and conducts away from the synapse, and the synapse is the joining. So the synapses can go from the axon of one to the dendrite of another, or you can go from the axon of one to the body of another, or you can go to axon or dendrite to dendrite, but those are less common. So this is axon to dendrite, this is axon to body here. We can have things called electrical synapses. These aren't very common. These are basically gap junctions. We see this kind of electrical synapse between the cardiac muscle cells in the heart, in the intercalated discs. It happens in some regions of the brain, but it's not very common, so we're not going to spend any time looking at it. Mostly we're looking at the chemical synapses, and this is exactly like the neuromuscular junction, where there is the presynaptic neuron is going to release a neurotransmitter, a chemical, into the cleft, into the gap, and there will be receptor regions on the postsynaptic cell, in this case another neuron. The cleft is a fluid-filled space, and that separates the two. So there's a chemical event that happens across the cleft. The neurotransmitter is released by the one, and there is receptors on the other cell membrane. And this means that it's a unidirectional communication. It only goes from the presynaptic to the postsynaptic, this way. So when the neurotransmitter diffuses and binds to the receptors, which are probably chemically gated channels, something happens on the postsynaptic. Now, in the neuromuscular junction, the acetylcholine opens sodium channels and becomes excitatory. But in nerves, we can also have these receptors close channels and make it more difficult, so they could be inhibitory. This will look exactly the same as what we talked about in muscles. There is the neuron and with the vesicles and calcium channels, and it's exactly the same as we talked about. So an action potential reaches the neuron, voltage gated calcium channels open, and calcium floods into the axonal terminal. The calcium binds with the vesicles that have the neurotransmitter in them that causes them to go to the surface and open, releasing the neurotransmitter into the cleft. The neurotransmitter in the cleft binds with the receptor, opening channels. Then the neurotransmitter has to be either taken back into the cell, into the presynaptic cell, or diffuse away or be broken down. In the case of acetylcholine in a neuromuscular junction, it's broken down by acetylcholine esterase. Serotonin is a neurotransmitter that gets reused, so it gets retaken up by the presynaptic neuron. Hence, we have a class of drugs called serotonin reuptake inhibitors that slow that down, which means there's more serotonin stays in the cleft, and the synapse becomes more sensitive then. That's kind of all of that together. So we either degrade, reuptake, or diffuse away. Degrading is quick. Reuptake means you get to reuse it, and it's fairly quick. Diffusion takes longer. There's a time delay because the neurotransmitter, all these things take time. So it's usually the rate limiting step in any neural transmissions. So we can have excitatory postsynaptic or inhibitory. And the strength is determined by how much neurotransmitter is released and the amount of time the neurotransmitter is in the area. So if we inhibit the reuptake, there will be more neurotransmitter in the area for longer. And so you will have a stronger stimulus. So we're not worried about graded potentials. Okay, so what we talked about in muscles was an excitatory synapse. So it's an EPSP. So the neurotransmitter binds, open the channels, sodium flows in. So sodium going in is greater than potassium going out, so you have a depolarization, and then the excitatory synapse potential, the EPSP triggers the action potential as long as it reaches the threshold and opens the voltage gated channels. If it's excitatory but doesn't reach the threshold, then you don't get an action potential in the postsynaptic. So the excitatory postsynaptic potential isn't strong enough. So this one didn't reach the threshold. Inhibitory, the neurotransmitter binds to and opens channels for potassium. And then that causes a hyperpolarization. And we remember the hyperpolarization moves you away from threshold, so it's more difficult to reach threshold. So it actually inhibits the postsynaptic neuron's ability to have the action potential. So that's why it's an IPSP, inhibitory postsynaptic potential. And it causes hyperpolarization. Usually, just a single excitatory postsynaptic potential can't induce an action potential in the postsynaptic neuron. But add them up, and you can summate them, and you can reach the threshold. But inhibitory ones will also summate, and they will cancel out. So you need enough excitatory ones to reach the threshold. So say it takes 10 excitatory to reach the threshold. You get 10 excitatory, you reach threshold. If you have 10 excitatory and 1 inhibitory, you don't reach threshold. You need 11 to counterbalance that 1. Etc. So neurons are stimulated by both excitatory and inhibitory, often at the same time, and the summation is how you reach threshold. Now the summation can come either from the same neuron firing one after another, and so that the excitatory happens from the same neuron over time, and that's called temporal. Or the excitatories can come from a number of neurons synapsing to the postsynaptic neuron, and that's called spatial. So if you've got a friend that wants you to do something, make a phone call. Their friend can say, "Come on, call. Did you make that call? Look, you're just wasting time. Make that call. Make that call. I'm not kidding. You really should make that call. Make that call. Make that call." That's temporal summation. But if you have 10 friends each say, really you should make that call, and each one of them says it once or twice, then that is spatial summation. These are just charts of that. So if you use the synapse often, you get an increase in efficiency of the neurotransmission, because calcium concentrations increase, and it just becomes better, and you become better at releasing more neurotransmitter because of that. So, we'll just move on from this. So, neurotransmitters. There's 50 or more chemicals that act as neurotransmitters. We can classify them either by what type of molecules they are or by their function. So, acetylcholine is a very important one. We've already talked about it in muscles. It's the neuromuscular junction, and it is some autonomic nervous system ones. So it's synthesized in the axon, in the presynaptic axon, and it's degraded in the cleft by acetylcholine esterase. We can have biogenic amines, so catecholamines. So this is things like dopamine, epinephrine, norepinephrine, serotonin, histamine. These are they play a big role in behaviors. So epinephrine, norepinephrine are involved very much in the sympathetic nervous system, whereas acetylcholine is the parasympathetic. Serotonin is the happy one. Histamine is a vasodilator as well as a neurotransmitter. And they're based on nitrogen, and that's why they're called amines. Amino acids can be neurotransmitters. So GABA, which is gamma aminobutyric acid, is one. Glycine, aspartate, and glutamate are important ones as well. Glutamate tends to be in the central nervous system an excitatory, where GABA is an inhibitory. This is why people become gluten intolerant, because gluten has a lot of glutamate, and you will so it affects the brain. It affects regions of the brain because it acts as a neurotransmitter. Dairy products have a lot of glutamate in them as well. But so do flavor enhancers, so monosodium glutamate. Almost everything that is low fat has added glutamate in it so that it doesn't taste like cardboard, because it tricks the brain into thinking, oh, this tastes good, because a good taste excites the brain, and glutamate excites the brain. So if you got something that doesn't taste good and you add glutamate to it, you think it does taste good. Glycine and aspartate do similar things. This is why they use it for sweeteners. So those are just plain amino acids. There are peptides, which are like small protein-like things. So endorphins and substance P, etc., fall into these classes. There's small polypeptides. You can have purines. ATP can act as a neurotransmitter. There's various fats that can do it. Nitric oxide is a big one. The endocannabinoids are lipid soluble. They're synthesized on demand. They're involved with learning and memory. They're cannabinoids because they're the same, they're similar to what's found in cannabis, which is why if you are ingesting cannabis, you feel that you have great insights and you know, things take on much larger proportions because it will stimulate the receptors of these neurotransmitters. So we're not going to get into too much into the functional classifications, but GABA and glycine inhibitory, glutamate excitatory, acetylcholine excitatory, and there will be receptors postsynaptically for all of these things, or for a lot of these things anyways. So they can act either directly on the channels, or they can indirectly bind to G protein, and it's almost a second messenger sort of thing, and it activates changes within the postsynaptic. So it's a much longer lasting effect, whereas the opening a channel is only a very short effect. So there's the channel linked and there's the G protein linked ones. And we will end this video right here.